Okay, so welcome to this next video. Okay, so what we're going to do in this video is summarise the adaptive immune system. Now, this is to be intended to be a video that can be watched at the end of the 11 videos on an introduction to the adaptive immune system. Alternatively, it could be used as a revision video. It's not intended to be a gentle introduction to the subject. I have 11 videos entitled Introduction to the Adaptive Immune System, where we draw all the pictures that I'm going to show you now out step by step. Okay, but now we're going to summarise the process. Okay, so there are two main forms of the adaptive immune response. There is the cell-mediated adaptive immune response, which is intended to attack pathogens which are infecting individual cells, so they are actually within the cells of your body. So the cell-mediated adaptive immune response is going to be against intracellular pathogens, whereas the other branch of the adaptive immune response is the humoral, which is intended to fight pathogens which are extracellular. So these are pathogens that aren't actually living within cells, but instead are living in the tissue fluid next to cells. So these are living in the tissue fluid, living in the interstitial fluid around our cells. So they are an infection of the meta-organism, whereas these are actually an infection of the individual cells. Right, so let's start off the immune response to the humoral attack, so the immune response that we're going to initiate to an extracellular pathogen. Okay, so what will happen is professional antigen-presenting cells will start this process off, and the main professional antigen-presenting cell that starts this process off is the dendritic cell. But we've drawn all the pictures with the macrophage simply because it's easier to draw. But both the dendritic cells and the macrophages are professional antigen-presenting cells. Now, they will phagocytose this extracellular pathogen into a phagosome, and they'll break it down. And many of the proteins that are on the surface of this extracellular pathogen will be broken down into fragments, and those fragments will be presented on major histocompatibility complex class 2 protein complexes on the surface of the antigen-presenting cell. Okay, so we call this an antigen fragment. An antigen is just any protein on the surface or otherwise any protein that is associated with the pathogen which we can launch an immune response against, which we can build an antibody against. Okay, right. So then what's going to happen is this professional antigen-presenting cell is going to go into the lymph vessels, and it's going to drain to a lymph node. Then what's going to happen is when it arrives at the lymph node, it will percolate through these two layers of the lymph node, the cortex of the lymph node and the paracortex of the lymph node. Okay, so this outer bit of the lymph node here is what's known as the subcapsular sinus, and the fluid that's coming in through the afferent lymph vessels needs to get from the subcapsular sinus to the medullary sinus at the center of the lymph node. So it has to drip through the layers of cells which are in the lymph node, which are the cortex, this outer layer with the B cell follicles in turquoise here, and then the paracortex, which is in green here. Okay, so the cortex and the paracortex are absolutely filled with T lymphocytes, okay? And there are two major types of T lymphocyte. Okay, so let's go over onto the next page. So the two major types of T lymphocyte are CD8 positive T lymphocytes and CD4 positive T lymphocytes. Now, CD8 positive T lymphocytes are not important for the humoral immune response, okay? So this response to the extracellular pathogen, that we're not going to use the CD8 positive T cells. So we care about the CD4 positive T cells, which are in the cortex and paracortex of the lymph node. Now, CD4 positive T lymphocytes have CD4 on their surface, as well as a T cell receptor. Now, you will have absolutely loads of CD4 positive T lymphocytes, okay? And all of these CD4 positive T lymphocytes will have different T cell receptors on their surface, okay? So the T cell receptor will be nearly identical, but this little binding site here, which binds to a fragment of an antigen, okay, this will vary. It will be different for every single CD4 positive T cell within the lymph node, basically. Okay, now that variation is a very important concept because they will all bind to different antigen fragments, basically. So, here comes our professional antigen presenting cell, which has the antigen fragment presented on major histocompatibility complex class 2. Now, it's going to have to find a T cell that has a T cell receptor which will bind to this antigen fragment on the major histocompatibility complex class 2. So it will have to search long and hard to find this, basically. It's not just going to find it instantly, it's going to take a long time. You're probably going to have to bring a huge number of different antigen presenting cells in, which will all have the same fragment on their surface before one of them will actually be lucky enough to find a T cell which is a match, basically, which has a T cell receptor which will bind well to this antigen fragment. Okay, but when you have found this CD4 positive T cell with this complementary T cell receptor, the antigen fragment presented on the major histocompatibility complex class 2 will bind to this T cell receptor here. Okay, then you've also got the CD4 molecule in green here, and this will bind to the major histocompatibility complex class 2. And this is important because this is why these antigen presenting cells which have antigen fragments mounted on MHC class 2 cannot activate CD8 positive T cells, because CD8 does not bind to MHC class 2, it binds to MHC class 1. Okay, so you have to activate the CD4 positive T cells. Now, the binding of the T cell receptor and the CD8 to this MHC class 2 complex with this mounted piece of antigen is going to give the CD4 positive T cell signal 1. Okay, however, that's not enough to activate the CD4 positive T cell yet. In order to activate the CD4 positive naive T cell, what you need to do is also give it signal 2. Okay, now this involves the professional antigen presenting cell putting co-stimulatory molecules on its surface, such as CD40 shown here in orange, and either B7.1, which is also known as CD80, or B7.2, which is also known as CD86, which is this red one here. Okay, now this professional antigen presenting cell, which remember was either a dendritic cell or a macrophage, this will be activated to put CD40 and CD80 or CD86 on its surface by the activation of its pattern recognition receptors. So remember, dendritic cells and macrophages have a whole bunch of what are known as pattern recognition receptors, PRRs for short, which recognize molecules that are associated with pathogens which human cells would never ever make. And these molecules which pathogens have, which human cells would never ever have, are a dead giveaway that there is a pathogen basically in the vicinity of this antigen presenting cell. So these molecules are what are known as PAMPs, which stands for pathogen-associated molecular patterns, and they activate receptors that the professional antigen-presenting cell has, known as pattern recognition receptors, PRRs, and this leads to the activation of the antigen-presenting cell so that it puts these co-stimulatory molecules on its surface. Now you might wonder, well, what's the point of this? Well, the point of it is it's a checkpoint, basically. It's making sure that this professional antigen-presenting cell is not presenting you a self-antigen fragment. You don't want this to be a portion of your own proteins, basically, and there's a risk that it could be. Okay, so these co-stimulatory molecules are only going to be on the surface of the antigen-presenting cell if the antigen-presenting cell has also been activated, i.e. if it's detected PAMPs in its vicinity, basically. So it's a checkpoint. It checks that there really has been a pathogen in the vicinity of this antigen-presenting cell that is bringing this antigen fragment to this CD4 positive naive T cell. Okay, so the co-stimulatory molecules bind to receptors on the surface of the CD4 positive naive T cell, and these receptors are CD40L, or CD40 ligand, for CD40, and CD28 for either B7.1 or B7.2. Now, when CD40 ligand and CD28 bind to those co-stimulatory molecules, that delivers signal 2 to the CD4 positive naive T cell, and once the CD4 positive T cell has had both signal 1 and signal 2 delivered to it, that causes the production of two things within this naive CD4 positive T cell. It causes the production of interleukin-2 receptor alpha. Okay, so the interleukin-2 receptor is a trimer. It's made up of an interleukin-2 receptor alpha, an interleukin-2 receptor beta, and an interleukin-2 receptor gamma. Okay, and basically, usually, these naive CD4 positive T cells will have on their surface beta and gamma components of this interleukin-2 receptor, but they won't have the alpha, so it's incomplete basically, and it's unfunctional, non-functional. Okay, but now they will start producing the alpha portion. This will bind with the already dimerized beta and gamma portion to create you a full interleukin-2 receptor. In addition, the CD4 positive T cell will produce interleukin-2. So basically, this looks rather futile, because what's going to happen is that it's going to produce the interleukin-2, secrete it out, then the interleukin-2 will bind to the interleukin-2 receptor and activate that T cell. So this self-stimulation is known as autocrine signaling. And once the interleukin-2 has bound to the interleukin-2 receptor, that will deliver signal 3 to the CD4 positive T cell. And that then will cause this CD4 positive naive T cell to activate. Okay, now this bottom picture here is relevant to CD8 positive T cells, so we'll come back to that when we discuss cell-mediated immunity. Okay, so we'll continue with this discussion in the next video, where we'll see what's going to happen to this activated CD4 positive T cell.